Well, the thing that started me in research is that when I went to college, I just fell in love with science, in particular with molecular biology. I like things, all things DNA and RNA and cells, and so I got very interested in molecular biology. And then I went to graduate school. Okay, I'm going to tell you the real story. So I met my husband as an undergraduate. We were both post-baccalaureates for one semester, and he had already been accepted to graduate school, and I was applying to graduate schools. And we were partners in an electron microscopy lab. So we fell in love, and so the deal was if I got into UC Santa Barbara, which is where he was going, that we could get married. And if I didn't get in, then I was just going to go to wherever I could get in, because I felt like I couldn't be happy watching him do what I wanted to do. So since I have a PhD and we are married, clearly, I did get in. And so we did our undergraduate works in completely different areas. So I'm trained as my PhD is in biochemistry and molecular biology, and his is in neuroscience, and he studied specifically color vision. So when we were in graduate school, during that time, there was another graduate student at Stanford who had, as part of his thesis, cloned the cone photopigment genes that are involved in the cone pigments that are involved in color vision. And I was really fascinated by that because, and I have color blindness in my family. So we spent a lot of time talking about color vision at home over dinner and breakfast. And when it came time to do a postdoc, we're trying to figure out what to do, because trying to find a place where both people can go and develop their careers further is somewhat difficult. So my husband said he wanted to stay in the lab he had done his PhD in, which is a color vision lab, and so I said, well, there's a lot that you could understand about color vision and color blindness if you could, apply some molecular biology to this field. So that's what we did. So we started collaborating as postdocs at UC Santa Barbara. And that's kind of what launched my entire career in this whole color vision thing. So the thing is, if you're interested in developing therapies for blindness, which I am, you have to pay attention to what the FDA requires if you're going to actually get something to where you're testing it in humans. So the way that the sort of standard things that people do is they try to find an animal model of the disease, and for humans, for a human disease, it's better to get a larger animal model, and the closer you can get to humans, the better. But it's really hard to find those animal models. People are beginning to find non-human primates now, macaque monkeys, that actually have diseases that are counterparts to what humans have. The challenge with doing anything in a primate model is that they have gestations that are like humans, so it takes a long time. And it's really expensive. And there's sort of some cultural pushback to doing primate research. So there are dog models of research that are larger animals, but people began by doing some really big animal models, like labor congenital amaurosis was done in the first treated animal his name was Lancelot, and he's some sort of giant dog breed, really cute but huge. But the dog world, like everyone else, it's expensive to have a huge animal, so they've all gone to trying to breed these into other kinds of strains that are much smaller. So, and then mouse and rat models, rodent models are very common because they're genetically manipulable. So those were some of and I've worked with all of those models. I've worked with mice, gerbils, macaque monkeys, squirrel monkeys, old and new world primates, and then we do a lot of work on human patients, human subjects as well. So if you're going to do some sort of treatment, like if we're interested in doing gene therapy, we basically have to have an animal model of the disease that we're trying to cure and try to show efficacy in that animal model. Once you've done that, then there's some reason to go on. But then you have to show safety. You have to show that, like, if you inject your virus into the eye of an animal and it suddenly has an adverse event, like it goes blind in that eye, you want to know whether that is because your virus did something, or the gene you injected did something, or if that was just the natural history of the disease. So people do the safety studies typically in mice, but sometimes they do it in non-human primates as well, but they're not animal models of the disease, they're just showing safety that you can inject it in the eye and nothing bad happens. And then at that point you can go to humans. So the biggest problem is that the human eye is larger than most other animal model eyes, even the large animal models. And it's slightly different. So the circuitry is very primate specific. Humans and primates share the same circuitry, but none of these other species do. And so we have very high acuity that we share with the non-human primates, but rodents and dogs, their acuity is much lower than ours. So their circuitry is not as developed for reading. so that's a problem. And part of the things that people have done, there's gene therapy research has been done on a lot of different diseases. And when we first came to Seattle, there was a company here that had just gone bankrupt, and they came and talked to us, and they said that they had tried 6 different products and they all failed. And so now their company was done. And they said that basically, even when they took one of their disease models to testing it in chimpanzees, it worked in chimpanzees, but then when they took it to humans, it didn't. And in every case, it could find a reason. And the reason was that humans were different from any animal model they tried. So this becomes a huge problem. So the closest that we have for humans is non-human primates in terms of the circuitry and trying to understand what's going on with the retina and what kinds of things you can do to try to save vision. But until you take it to human trial, you won't know if it's going to actually work. So I think all of the models have their value, and it's just a matter of what specifically you're trying to do. The last 5 to 10 years, one of the biggest things that has happened is that people have gone from doing really individual investigator initiated research to the NIH wanting to fund these larger consortium, collaborations between groups of people across, different institutions across the country, and get these, big name people all together working on the same problem on a cooperative project, and then spend a lot more money on it. So I think that has not worked as well as people had thought it would, and I think that we might see a shift back towards more basic and individual initiated research. I could be wrong, right? Because I have no ability to predict the future, but this is what I've seen from the last 5 to 10 years, is that this has been a pathway that people have tried, and it turns out it's difficult. And you know what I said at the beginning, the first question, one of the things I just love is the fact that I get to be in charge. But when you have a lot of big name people, that are all working together, nobody gets to be in charge. Or if you put someone in charge, then everybody just wants to let that person be in charge and report back to us, and we'll give you advice, but we'll go off and do our own thing that we're really interested in. So I think that as a model is a little bit difficult when people are remote. When everybody's all in the same institution, I think it works much better.